Hi, I'm Angela Hwang, CEO of Metaphore Biotechnologies. Thank you. What problem in antibody drug design led to the creation of Metaphore? Well, historically, the way drug design happens today is that you take a receptor of interest, you crystallize it, and you solve for binders to that crystal structure. The problem with that approach is that you're taking a static structure which has now been completely isolated from its endogenous environment, and what you have lost is the biological context and any information about the functional relationships between the receptor and its binding partners. So what we're trying to do here at Metaphore is to do the complete opposite of that. How can we replicate the living system? What that receptor and all of its binding partners are actually doing in its native state, and to use that as the starting point for our drug design. How does Metaphore actually turn living biological behavior into new medicines? The Metaphore drug design process has 3 steps. The first step is an experimental step where we recapitulate the activity level and the engagement between a receptor and all of its binding partners in its native state. We capture all of those interactions. Actually, we capture up to 100 billion of those interactions. Each of our data sets have 100 billion data points. And we put that into our next step, which is called Mimic. In the step Mimic, what we do is to apply our computational model to extract and understand of all of those interactions, which are functionally relevant and which are not. So we try to separate the signal from the noise. Once we know those interactions that are functionally relevant, we call that our digital blueprint. We then move that into our third step, which is called design. In the design phase, we take that digital blueprint of function, right? Because we know that that's where all the activity is happening. And we use that as the foundation to then rebuild the entire molecule towards the TPP, the target product profile of our desire. So we'll be looking for certain solubility parameters, we'll be looking for certain stability parameters, we'll be looking for long half-life. In this last step is where we use our proprietary computational models to then rebuild the molecule to its ultimate state. What kinds of medicines are possible now that weren't possible before? Well, today, most antibodies are called antagonists, which means that they work by blocking a receptor. Through the Metaphore platform and the ability for us to do function-first engineering, what we've been able to create is a whole new class of antibodies that are called functional antibodies. These antibodies are special because they open a whole new world for antibodies that were not possible before. We don't just block. What we're now able to do is to actively engage biological pathways and drive signaling pathways in ways that were not possible before. So we can agonize pathways, we can tune bias in a way that's very specific to prefer one signal versus another. We can engage multiple targets at the same time through a single paratope. In the format of a single monoclonal antibody, can now engage multiple targets. And we can even, with a single paratope, engage and create molecules that can do both antagonism and agonism at the same time. So what this means is that we have now created a whole new toolbox to engage complex biology and disease biology in ways not possible before. We can look forward to a whole new class of first-in-class as well as best-in-class molecules. Thank you very much.